we are going to introduce our method to establish a strain of algae, especially unicellular conjugatophytes. For the establishment of a strain, collection of water sample which contains algae through fieldwork is necessary. The water sample contains foreign matters other than algae. Only single cell is isolated from this sample. The cell is transferred to a clean medium and cleansed repeatedly. Isolation is conducted through this process. Sterilization is possible if it is conducted in a careful manner. Finally, the isolated cell is transferred to a test tube and the strain is established through cultivation. Here, we focus on unicellular conjugatophytes in which varieties of forms are identified. Unicellular conjugatophytes grow in stagnant freshwater area such as lake, marsh, and paddy field. From these environments, algae containing water sample is collected to produce a culture strain. We will introduce a method to collect water sample from lake environment. A plankton net is used for the lake environment. The upper net part is permeable to water.